So as Jackie said, I work at the Royal Botanic Gardens. I also collaborate a lot with UNSW. And just as you might tell from my name or accent, I'm previously from Paris. I worked in the university in Paris for many years before I moved here to Sydney 4 years ago. So the work that I will present to you today is a collaborative work with two Mexican colleagues, Santiago Ramirez Barahona and Susana Magallon, that we published just about a year ago. And so I just want to tell you a little bit about this story. Oops. So before I start, I'd just like to specify my research focuses on flowering plants broadly. This image was compiled by Santiago, showing a little bit of the floral diversity in flowering plants. And really what I'm really passionate about is this extraordinary diversity of flowers that we see today and how it might be linked with the diversity of species that we have in flowering plants. As you might know, 9 out of 10 species of plants are flowering plants. This hasn't always been the case, and we'll go a little bit into this today. But nowadays, it's a group of enormous importance in all terrestrial habitats. We wouldn't be here without flowering plants. So I'm interested in understanding how flowers have interacted with the diversity of flowering plants. And this links naturally into also questions of divergence times for the evolutionary timescale of flowering plants. So this is what I will be talking about today. And before I start, I'd just like to know that there are quite a few things that we don't know yet. And one particular aspect of flowering plant evolution that is critical for understanding of what happened is the actual age of flowering plants. So I'd just like to start my talk by telling you that today, in 2021, I believe we have absolutely no idea when flowering plants originated. There's been a lot of hypotheses, but nearly every 2 months, there's a new paper showing different results. So if you ask people from the paleobotanical community, there's a strong consensus that it is very unlikely that flowering plants originated long before the first appearance of fossils in the fossil record, and that is dated to no more than 130 million years. So before 130 million years, there is no equivocal fossils for crown group angiosperms that we know of the fossil record. And this particular figure is taken from a recent review by Pat Herendino in 2017. But at the same time, there's been an enormous amount of studies using molecular dating analysis with fossil calibrations. And depending on the constraints that were applied to the root node of angiosperms, some of these analyses have consistently recovered a much older date. So this is just an example from a work by Charles Foster and colleagues a few years ago, where we found an age ranging from 192 to 250 million years. So at the moment, there's still this complete unknown. We don't know when angiosperms originated. It depends on who you ask, but more importantly, it depends on the assumptions that are put in the analysis. And this is just a figure that we published 2 years ago to summarize that situation. So the crown group of angiosperms, which is formed by their most recent common ancestor and all of its descendants, living and fossil, that is what I'm talking about. That is the age that we're still not clear about. On the other hand, there's better knowledge about the divergence of angiosperms from their sister group, the living gymnosperms, the acrogymnosperms, and that is what we call the stem node. So the difference between stem and crown nodes that many of you might already be aware of, that is critical for the rest of my talk, and I'll get back into this a little later. So the question that I want to ask in this talk today is not the age of flowering plants, but rather the age of the numerous clades within the flowering plants, in particular the many orders and families that compose the flowering plants. And this question can be answered, or at least we're answering here, with a combination of fossil calibrations and molecular dating analysis. So there's been a lot of work on relaxed molecular clocks in recent years, allowing to use information from molecular trees, so the branch length, basically the amount of inferred change based on molecular data, transforming that into a timescale. But to do that, no matter which software you use, you need some external information. And usually the external information comes from the fossil record. And there's increasing evidence that the more fossil calibrations you have, the more accurate we may hope to have divergence times. And so this is my part in the picture. I've been working mostly on improving fossil calibrations in both quality and quantity. There's a precedent, however, in the realm of molecular dating analysis of angiosperms, and this was the groundbreaking work by Susana Magallon and her colleagues, published in 2015. So it was one of the dating analyses that included many representatives of flowering plants. In this case, about representatives from about 380 of the 420 families in flowering plants. But it really departed from previous work in the number of fossil calibrations included. There were 136 different fossil calibrations in that analysis, whereas every other study was on an average from 20 to 50 calibrations. So what we wanted to do here with Susana and other colleagues was to go even beyond what was done back then. And to do this, I developed a particular part of a database that I've been working on for a long time for morphological traits. So I developed a calibration module, and I'm just showing you here a little screenshot of what it looks like. And basically what we try to do here is go deeper into justifying every aspect of a fossil calibration, including what it is, so the fossil identity, who described it, citing specimen numbers, in which formation it was found, but also its relationships, which sometimes forces us to go look into completely different literature, either recent reviews of calibrations or recent phylogenetic analysis. And lastly, we also reviewed the age of each fossil, which may have evolved since the original description of the fossil. So I won't go much more into detail in this example, but in doing that, we were trying to follow the 5 best practices proposed by Jim Parham and all in 2012 to justify fossil calibrations. But actually, we took that even further. So we added more information than they suggested originally to do. And so what we did is we took a whole bunch of recent and older papers. So we took the big list of calibrations from Magallon and all, but we also supplemented that with recent reviews of fossil calibrations. So these papers might not speak a lot to you, but this was a whole review of all the monocot calibrations, a review of all the asteroid calibrations, and a review of magnolid calibrations. And then we also integrated more recent papers that have published very interesting fossils that definitely have a potential for calibration. We put that into the database, PROTEUS, and this led us to look at more than 300 fossil calibrations. And so for each of these, we tracked the original descriptions, sometimes having us look into very old literature in different languages. And our primary rule was that if we could not track the original description, we did not include the calibration. And so this was to solve a problem that appears a lot in molecular dating analysis. There's a lot of recycling of calibrations from one paper to the other, and sometimes the information is lost or the original intention is lost. So here we try to always go back to the original source. Then we reviewed the node assignments for each of them and also the ages. And we did that multiple times to also align calibrations together. So if they came from the same sediment, we made sure that we were using the same ages for these calibrations. So after excluding some that we deemed unsuitable to use, we ended up with this new fossil calibration database for angiosperms of 238 fossils. So the goal of using these calibrations was to date again the timescale of flowering plants. And this is where Santiago came into the picture. He assembled a molecular tree using, I think, about 7 genes from traditional Sanger sequencing, but putting all of the gene bank data together for all of the families of flowering plants. So where this tree was really unique compared to any other tree that had been published back then, it is the first tree that includes all of the families that we currently recognize. So in our system, this was 435 families, and of course, all orders. And so this tree included 1,200 taxa. And we calibrated it with 238 fossil calibrations, applying to 210 distinct nodes. So some calibrations would apply to the same nodes. Sometimes this happens. And we also analyzed the subsets, restricting ourselves to just the calibrations that have been actually put into phylogenetic trees. So I'll explain a bit later why that is important, but basically the results were very similar if we just restricted our analysis to that. And so this is just the tree here on the right-hand side. It shows you the distribution, the phylogenetic distribution of our calibrations. So the black and white dots are all of our calibrations. There are still some clades for which we have very few fossils, but overall we had calibrations all over the tree. So there's good evidence that when molecular rates are very heterogeneous through time and across lineages, the more calibrations, the better. And now, before I show you just the key result from that study, I should mention that this tree was not only unique in having all of the families, but even more importantly, for most of the families that have more than 2 species, we specifically sampled the crown nodes of these families. So we chose representatives from living members so that according to current phylogenetic knowledge, we would represent that crown node in our tree. So that allowed us not only to quantify the stem nodes for each family, but also their crown nodes. And I'll show you just a couple of examples from like the concrete examples from the flora surrounding us. So this is a particular species, Eupomatiaceae lorina, that belongs to a genus and a family, Eupomatiaceae, that only has 3 species, predominantly distributed in Australia. And for those who know me, I'm passionate about this particular family. And this, in our analysis, because we sampled the crown node, we were able to estimate the divergence of the 2 main species of Eupomatiaceae at 25 million years, with a wide confidence interval, as you can see, 4 to 82 million years. And on the other hand, their divergence from their sister group, the very large family Ananaceae, was estimated at 98 million years, or 83 to 113 million years. So what's important here is that we have 2 very different ages for each group, and there can be a very long difference between these 2 ages or a very short one. It depends on the group. So in the case of Eupomatiaceae, the divergence among living species is very recent, which also serves to illustrate an important point, is that this particular flower has been portrayed by some people as a very ancient relict, a member of things that happened all the way back to the Jurassic, if you listen to some stories. The reality is that the species is no older than actually 25 million years. So it's very important to consider these 2 different ages when relating to the flora that surrounds us. Not everything is as ancient as sometimes we might want it to be. This is another example. This picture was also taken at the Royal Botanic Garden in Sydney, but that flower grows in northeast Queensland. It's a member of the small family Calycanthaceae, and it is Angiosperm australiense. It's a beautiful flower. And here we date the divergence of Calycanthaceae, the whole family, as a much older family. You see the crown node here is 108 million years on average. And the stem node is a little bit older, but not as older than it was for Eupomatiaceae. So we calculate a difference here of, on average, 23 million years. So we did that for every family in flowering plants. This is the last example, Proteaceae, another group that I'm very interested in. Here we have sort of an average fuse difference of 37 million years between their stem and their crown age. That's the family, by the way, of Waratahs and Banksias. So if we just summarize these results over all of the families that we had in our sample, we found that most of the family stem ages were distributed in the Cretaceous. In fact, 59 to 80% were in the Cretaceous. There's some stem ages that are a bit younger, and possibly some that are older, depending on our constraints on the root age of angiosperms, but most of them are in the Cretaceous. And this fits well with the narrative that a lot happened in the Cretaceous. There was an enormous diversification of flowering plants. We see that in the fossil records. It's been known for a long time that the Cretaceous has been the key period for radiation of angiosperms. But interestingly, if we look at the crown ages, we now find that most of these are actually in the Cenozoic, post the KT boundary. And this is very important because this is actually when most of the species diversity that we see today has been produced. And by measuring the crown nodes, we look at the most recent common ancestor of all living representatives. So the sort of beginning of modern diversification of the families. And to be honest, we were of course expecting that crown ages would be younger than stem ages. But there was really no previous thinking about whether they would be much younger or just a little bit younger. And here we find a huge difference. And I think that means we're still trying to think what it means, but it probably is telling us something either on flowering plant diversification or on the way that botanists have been delimiting families for a long time. In fact, when we look at the difference, the differences are variable, according to the families that you look at. But on average, there's 40 million years to make up a family of angiosperms. That's the average difference between the stem and the crown node of all of the families in our sample. What's really interesting is that there is growing evidence from both paleobotanical literature, but other sources of evidence as well, that in fact, it might be a misconception that everything changed in the Cretaceous for angiosperms. And in fact, there's a lot of evidence that shows that angiosperms only became dominant ecologically across the world in the beginning of the Cenozoic. And so the story that we get from dating actually kind of fits the new narrative from paleobotany, that angiosperm diversification didn't all happen in the Cretaceous, but there was a major phase in the Cenozoic. And this is probably one of the most interesting results that is emerging from that study. So just to wrap up, I'd like to show a few results from more recent work that we're doing at the moment. And this is the so-called Paleo eFLOWER project, which I've been running with colleagues in Vienna, Jörg Schönenberger and Maria von Balthasar, for quite a while now. And so what we're trying to do here is use a framework of floral trait data that we have built for flowering plants to better understand the fossil record, and specifically put the fossil record onto the phylogenetic tree of flowering plants, and ultimately try and understand what is this telling us about floral evolution. So a few years ago, we published a model, a reconstructed model for the putative ancestral flower of all flowering plants, and also putative ancestors for all the key chelates of flowering plants. So to do that, we assembled a data set of 800 species and 27 floral traits that we recorded and then reconstructed using ancestral state reconstructions. This particular tree was a reconstruction of the sex of the flowers. So what we've been doing here in the Paleo eFLOWER project was to use this data set as a morphological data set to run phylogenetic analysis of fossils. And I'll just show you this example of a fossil from the Cretaceous of Germany, Moldinia, that has been attributed classically to be a member of Lauraceae. And for those who know the family Lauraceae, that's the avocado family, by the way, the flowers, the reconstructed here, are very similar to modern Lauraceae. So it's no surprise that paleobotanists have suggested this was probably a member of Lauraceae. And when we run an analysis here, putting this fossil in this big data set and allowing the fossil to go anywhere in the tree using a so-called molecular backbone analysis here with parsimony, we find that actually the most parsimonious positions are indeed in Lauraceae, or possibly on their sister groups. So this is a stem lineage of two other families, Hernandiaceae and Monimiaceae. So this is very reassuring, because in these analyses, for the first time, possibly ever, we are allowing the fossil to go absolutely anywhere in the tree of flowering plants, rather than constraining any analysis a priori by already assuming it's maybe a member of Lauralis and then conducting a Lauralis scale phylogenetic analysis. So we've been doing this for many fossils, and watch this space, because we now have a PhD student, Andrea Lopez Martinez in Mexico, who's putting all that together. With the ultimate goal, we hope to run a tip dating analysis with as many fossils as possible included as tips in the phylogeny, to better understand divergence times in flowering plants, but also the impact of fossils on flowering plant evolution. So thank you all for your attention.